Well, today's speaker is well known to everyone. Chris Emerling is an NSF postdoc in the MBZ working in my lab. And he arrived last year. This is his second year. He did his PhD with Mark Springer. And he gave a seminar at the start of last year. And he's setting a wonderful example by giving another talk a year later on a completely different topic. So as he said to me just a minute ago, you know, this is good for getting feedback and promoting interactions. That's what we're all about. So I won't say any more. I'll give the time over to Chris, and the title of this talk is there. So take it away, Chris.
Thanks, Michael. Yeah, because it has been almost exactly a year since I last came here and presented to you guys. I just want to say this last year has been completely wonderful. Just interacting with everyone. I've gotten so many good ideas. I've met so many kind people, and I just want to say I'm looking forward to another year, but perhaps more than that, depending on how funding goes after that.
Maybe we can turn up the lights just a little. Maybe leave one of those on. So yeah, I think that's better. Okay. If you need it darker, just say so.
Okay. So it's a bit of an understatement to say that light has an important impact on life, beyond the obvious of providing perhaps definitely the primary source of energy for life, and also in terms of how it influences the seasons, and so the availability of food, where animals are living due to migration, how animals actually use this light differs, and so some will use it to communicate with one another, some will use it to kind of hide from prey and predators. And the thing about light, when considering how organisms are using this and what's available to them, is light is not uniform across the Earth. So for instance, if you were to go to the ocean, and you go to the surface of the ocean, there's a certain intensity and a broad range of wavelengths in terms of spectral intensity as well. But as you go deeper and deeper in the ocean, light not only becomes less intense, it also tends to blue shift. Okay? When you go into freshwater environments, the opposite tends to occur, and so you actually get a red shifting of that light. And even in terrestrial environments, so just through the passage of day, you can see there's obviously a change in intensity. We all know that nighttime is dimmer than the day, obviously. You can see how great that intensity difference is, even between a full moon and starlight, but also even the spectral quality changes throughout the day. And so you might imagine, and we do know this from a lot of previous research, that how species adapt to different light environments differs. And the main way that organisms, specifically vertebrates, are taking in that light is through the eye. And the eye is a complex structure that doesn't just take the light as it is, it actually modifies that light. So what it does is it collects that light, it changes the intensity of how much light is available to the eye through, for example, the pupil. It focuses that light with the cornea and the lens. And then even as that light transmits to the retina, where light is absorbed, a lot of that gets filtered out even further, which I'll talk about more in a bit. So once we get to the retina, the business end of photoreception, where light is actually reaching, and so you might imagine some of the adaptations at the photoreceptor level taking place, you have light coming in from the bottom here. It goes through these multiple neural layers, and it's ultimately collected in these photoreceptive cells towards the back of the eye. Now there's two general types of photoreceptor cells in vertebrates. You have the rods, which are shown here being kind of thin and dark. These are involved in dim light vision, and the way they're specifically adapted for dim light is through multiple mechanisms and modifications to their structure, including just the overall structural, ultrastructural features of the photoreceptor allow for more photoreception. The kinetics for how quickly light is transduced into electrical signal is very sensitive, and they also tend to synapse onto multiple cells with synapse onto a single neuron, allowing for a sort of pooling effect, allowing for a greater dim light sensitivity. Cones do the opposite, so they're involved in bright light vision. So basically everything I just mentioned about rods, the opposite occurs in cones. So these are actually, they need a higher threshold in order to activate. And so as a rule, in bright light conditions, cones are what's functioning, in dim light condition, rods are what's functioning. Now cones actually also contribute to color vision, so you can kind of see how these are colored differently. This is due to differences in pigments that are present in various types of cones, whereas rods only have one type of pigment. So having different kinds of pigments, which absorb different wavelengths of light, allows for a comparison and contrasting of these different chromatic signals, allowing for color vision. So where light absorption actually happens at the molecular level is in these pigments that I was just talking about, which are composed of an opsin protein, which is a G protein coupled receptor, shown right here, as well as a chromophore, a vitamin A-derived molecule that's actually doing the light absorption directly. And the interaction between certain amino acids in this opsin protein and the actual chromophore itself leads to differences in spectral sensitivity across these various pigments. And so you can see right here, these are examples of human visual pigments. And you'll notice that they differ in terms of their spectral sensitivity, ranging from blue to greenish to reddish. And you'll see here we have a rod pigment in the dotted line, and then we have 3 different cone pigments, which are shown in the different colors. And again, by having these different cone pigments, different cone channels, allows for comparison of different spectral qualities and therefore color vision. Now, this is a human, but the ancestral vertebrate, we think, actually had 5 visual pigments. So an extra cone pigment specifically. And so birds actually represent that typical complement of visual opsins. I'm not showing the rod opsin here, but you can see here there are 4 different cone pigments. And so we have what's called trichromatic color vision, allowing for, again, 3 different color channels. They have tetrachromatic color vision, as a rule, birds do, allowing for 4 different color channels. So they have an extra dimension, so to speak, to their color vision. Now, thinking back again to differences in light environments and how species might be adapting to them, you can imagine, depending on the light intensity or the spectral quality, that there species might be adapting by evolving such that their number of visual opsins has changed. So you might imagine a reduction or a gain of new visual pigments. You might also imagine that the spectral tuning of these pigments would change. And so again, that can happen through changes in these key amino acids that are involved in spectral tuning, as well as a mechanism involving the chromophore, which we'll talk about later. So this brings me to the main focus of this talk, thinking about this particular group, Archelosaurian. For those of you that aren't up on your Sauropsid taxonomy, this is actually a very new term, and it is encompassing 3 general clades. So we have aves and crocodilians, which are known as the archosaurs, and turtles, which have at least historically been called chelonians or testudines. So recent analyses have suggested that this clade is in fact a real clade, that these all share a common ancestor to the exclusion of all other sauropsids. For those of you familiar with turtle taxonomy, you'll probably be aware that this has been actually controversial for quite a long time. They have an anaptid skull condition, which is thought to be primitive for amniotes. And they have very derived postcranial morphology, suggesting it's just really difficult to have placed them into a particular part of the amnio tree. But with new recent phylogenomic analyses, it's become quite clear that it's a very short branch. Nonetheless, turtles come out with birds and crocodilians pretty consistently. And you can see here's lizards and mammals as outgroups. And very strong support. And on top of that, well, what that implies is that turtles are therefore diapsids. So they have a diapsid skull condition that was somehow lost. They're secondarily anapsid, losing those temporal fenestrae. And sure enough, just last year, there was a stem turtle discovered with 2 temporal fenestrae, suggesting they are indeed diapsid sauropsids. So now, birds have been done to death. That's not entirely true. Birds are, there's still a lot more we can learn about birds, but I mean, in terms of visual evolution, birds, of course, are one of the premier groups to study. They're colorful and they're all diurnal pretty much. But turtles and crocs, not so much. And there are important questions about, or important aspects of their evolutionary history that probably would inform how their visual systems have adapted to different niches. So turtles, it's a little controversial what happened in the stem lineage leading to turtles, but for one of the earliest turtles shown here, Odontocheles, was found in marine deposits. This is a so-called half-shelled turtle, only had plastron, no carapace. And the clade that is most frequently recovered in cladistic analyses as being sister to turtles are the Sauropterygians, which are all, they're all extinct. Were all marine in their ecology. But there are also a lot of stem turtles that have very clearly have terrestrial adaptations. No evidence of anything aquatic going on. But at least what we can say, despite that ambiguity there, is based off the phylogenetic distribution of crown turtles, it seems very clear that freshwater ecology was something that they adapted to at some point before the crown ancestors of these taxa. And so we'd imagine that perhaps going from a terrestrial to a freshwater environment might have had some effect on the evolution of their visual system. And also, marine turtles, sea turtles, are an excellent example of a secondary reacquisition of a marine lifestyle. And this actually goes back pretty far, all the way back to the Mesozoic. So that also allows for another potentially interesting question. Now, crocodilians, they're all semi-aquatic in their general ecology, but the branch leading to crown crocodilians was actually full of a lot of various pseudoseuchian archosaurs with just disparate ecologies and different morphologies. You can see some examples right here. So that doesn't necessarily impinge on what we might expect to happen in the ancestor of crocodilians, but there is an aspect that goes all the way back to 1942 when Walls, who's basically the god of vertebrate comparative anatomy in terms of the eyes, he posited that perhaps the ancestral crocodilian might have been nocturnal. And not only was it nocturnal, but that it actually had this long period of nocturnal adaptation that led to an overall modification to the ocular anatomy. And so there's a few reasons to suspect this. One, a lot of crocodilians are nocturnal hunters. Their eyes tend to be rod-dominated. Crocodilians tend to have eye shine due to tapetum lucidum, so a reflective structure at the back of the eye that helps to collect light in dim light conditions. And on top of that, they've actually lost several features that we think the ancestral sauropsids and archosaurs had that are involved in bright light vision. So for instance, are these oil droplets, not coil droplets, these cone oil droplets. They're present in the cones of things like squamates and birds and turtles. And what they do is they absorb light that's actually shorter wavelength light specifically, that's on its way to the cones. So this is an example of a filtering mechanism beyond what actually comes to the eye. Okay? So what this does is it kind of sharpens those spectral sensitivity peaks, and what that allows for is greater color discrimination because there's less overlap between the different pigments. Right? The problem with these in dim light conditions is they absorb a lot of light. And so if you're a dim light species, you might imagine that these would get lost. And sure enough, these have been lost in crocodilians. Other features involve accommodation or focusing of the eye at different distances. If you look at the eyes of birds and lizards, you'll see they have, so here we're looking at a lens of the eye, cornea, here's the rest of the eye here, so kind of in a cross section. And you'll notice there's this thing called an annular pad. And this is a structure on which muscles are acting to help focus the lens. And crocodilians don't have this, whereas their sister groups and related taxa do have these. And then also these structures right here, sclerotic rings. So sclerotic rings are thought to help prevent distortion of the eye as the cornea is actually focusing the eye and keeping the image nice and fixed on the back of the retina. Crocodilians don't have these, birds do, turtles do, lizards do. And what's interesting about the loss of these features is that all seem to be associated with dim light. And so thinking about focusing images, when you're in dim light, you're not doing a lot of focusing with your eye. You're more concerned with sensitivity. So losing these features makes sense if you're dim light adapted. And some of these same features have also been lost in mammals. And so for those of you who might be aware, there's a hypothesis that goes back a long ways, that this idea that mammals were ancestrally nocturnal. And they lost some of these same features. The general idea behind it is they're living in the shadow of the dinosaurs, they're endothermic, and so they can be active at night. Whether or not that's a very complete story, who knows? But there are several features associated with bright light vision that have been lost in mammals as well as crocodilians. But on top of that, mammals have lost some aspect of their color visual system. They've lost visual pigments in their ancestry. And there are other genes associated with light reception that have been lost as well. And so we might expect that if crocodilians also went through a nocturnal bottleneck, similar to what we think happened for mammals, that their genomes would actually reflect this similar loss in light-dependent genes. So this is a genomic study, and what I did was I looked at 4 species of turtles, 2 soft-shell turtles, a green sea turtle as well as a painted turtle. You'll notice, for those of you familiar with turtle phylogenetics, these are all cryptodires. I don't have any plurodires for this study, so it does limit to some degree what we can infer about the ancestral turtle, but there are some things we can ultimately project backwards based off of the ancestor of cryptodires. And in crocodilians, I studied 2 alligators, a saltwater crocodile as well as a gharial. These represent the 3 families of crocodilians, so any changes, any evolution in that stem branch would represent what probably happened in the ancestral crocodilian. The genome coverage for these guys was generally very good, the one exception being the spiny softshell turtle, which wasn't great, but it was still good enough for what I was trying to do. And I looked for the 5 vertebrate visual opsins, as well as 12 non-visual opsins, so these are light-sensitive pigments that are involved in other functions, as well as 3 genes associated with UV damage protection, which I'll elaborate on later. I used the NCBI's genome annotation pipeline, as well as querying the whole genome shotgun contig database to look directly for the genes of these various species. And what I did for the latter is I designed these in silico probes, which encompass, so here's an example of a probe right here. It encompasses all the exons shown here, as well as introns and flanking sequence of the gene. That way it ensures that if a gene has become pseudogenized or deleted, that we're more likely to recover what's left of it. Align these, examine them for inactivating mutations, frame shifts, premature stop codons, etc. Perform some DNDS ratio analyses to test for positive selection as well as relaxed selection for specific genes. And I also did ancestral sequence reconstructions on spectral tuning sites. So these are amino acids known through mutagenesis studies and comparative studies to influence the spectral tuning of those opsin pigments. All right, so right to the results. So what I have here are the 4 turtle species of the study, and in increasing order of wavelength, I have the various vertebrate visual opsins. So these 2 are cone, these 2 are cone, the one right in the middle is a rod pigment. Okay? So the first thing you'll notice in the next screen is what has been lost. So in soft-shell turtles, some of the short wavelength opsins, or in this case both of them, have been inactivated or apparently deleted from the genomes of these 2 species, whereas these 2 taxa have retained all 5 pigments. There isn't much I can compare this to in terms of direct measurements of the retinas of turtles, but here's an example of one of the very few studies. And this is just looking at cone pigments and what they found. This is a red-eared slider, which is an amidid just like this guy. And just like I predicted with my amidid species, there are 4 cone pigments, and they all match up what you would expect based off of the opsin identities of these. So based off the presence of 5 opsins in the Durocryptodires, which are the green sea turtle and the painted turtle, it does suggest that 4 cone opsins and 1 rod opsin were present in the ancestral turtle. Okay? So what this means is tetrachromacy, so this dimensionality of color vision that's 1 step above ours, and we have in birds, is also was present in the ancestral turtle, as well as at least some descendant turtles. I did find on the stem branch for my taxon only one little element of evidence for adaptive change was positive selection on the red opsin. But nothing else. No spectral tuning sites. Like I said, no losses of particular genes, no gains of any genes. And there were several other spectral tuning site substitutions, one on the branch leading to sea turtle and painted turtle, and then as well as the descendant branches of these. And what's interesting, just kind of looking at this pattern, is SWS, these are the short wavelength sensitive opsins. There have been multiple spectral site substitutions, as well as losses associated with the shorter wavelengths. And this might be, just throwing this out here, this will have to be tested in more detail, but this might be because in freshwater habitats, like I mentioned earlier, the water tends to redshift wavelengths. And so perhaps there's something going on with short wavelengths, maybe not enough of that is penetrating, and so you either lose them or you have to modify them in order to absorb more light. This will have to be tested in more detail later, but that's one thing going on, potentially. And again, I didn't really find much in the stem turtle, or at least the stem cryptodire, which would, in some cases at least, you could infer about the stem turtle. No spectral tuning site changes, no gene losses. But there might be a different reason for why that is that has nothing to do with what I've talked about so far. Like I said, the way you can actually change the spectral tuning of your pigments is there are two different ways. One, you can change the amino acids that are present here in certain key tuning sites. The other way is actually changing the chromophore that's present in your pigment. And so vitamin A comes in two different forms, vitamin A1 and vitamin A2. And vitamin A1 tends to blue shift pigments. Vitamin A2 tends to red shift pigments. And what's interesting is when you look at freshwater fish, they tend to have vitamin A2, the red shifted one. When you look at marine fish, they tend to have vitamin A1, the blue shifted one. And again, not much has been done with turtles, but one study did actually look at this, and they compared just two species, unfortunately, but two species nonetheless. The red-eared slider which is a freshwater species, has vitamin A2, and the green sea turtle is a saltwater species, has vitamin A1. Okay? And so this suggests that perhaps rather than actually modifying the visual pigments in the ancestral turtle, if they indeed became freshwater adapted, perhaps all they had to do was shift which chromophore they had, which vitamin A molecule they had, and that was sufficient. Now, the only way to really get at this in more detail is to sample more species and just get an idea of what vitamins are present in their pigments, but that's the idea that I have going forward. Now with crocodilians, again, we're thinking about this idea that maybe crocodilians were ancestrally nocturnal, and we know when species have become nocturnal for a long period of time, they tend to lose visual pigments, cone pigments almost exclusively, because in bright light conditions, cone pigments are useful, and dim light conditions not so useful, right? And just like mammals, they lost two cone opsins. And first thing, besides that, you'll notice is they're very patriotic opsins, red, white, and blue. All four of the crocodilian species have been lost. So this gene right here, SWS1, appears to be deleted based off synteny information. I found no remnants of this whatsoever. This one right here, known as RH2, it is pseudogenized, so I was able to get fragments of it, and there's evidence of shared inactivated mutations in all four species, suggesting that it was indeed inactivated in the common ancestor of crocodilians. There is a potential frame shift in the Chinese alligator for the rhodopsin. That's extremely unusual, and all the other rod genes are intact, so I'm highly suspicious of this, but it'd be interesting for people to follow up later on. Now again, just like turtles, very little has been done in terms of measuring the pigments of crocodilians directly, but there have been 3 studies for which I can compare to. So one study looked at the American alligator, one looked at a caiman, and one looked at saltwater and freshwater crocodiles. So I actually have overlap with 2 of these species right here. And what they've found, so this is showing, so first they have found 3 general types of photoreceptor morphologies. So you have rods, you have single cones, and what we call double cones. So mammals don't have, well, placental mammals don't have double cones, but most other amniotes do. I won't get into their function because we really don't know what they're doing. They seem to be doing something with achromatic hues, but that's all I'll say for now. And what I have here below these photoreceptors are the direct measurements of the pigments. So they measure multiple photoreceptors, and these are the lambda maxes or peak values that they got. And so based off of the genes that are present in their genomes and these photoreceptors, we can assign what the likely pigments are being expressed in these different photoreceptors. So in the rods, the rod opsin is probably being expressed. It makes sense with the spectral profile as well. There's a single cone that matches very much what we would expect from SWS2, a short wavelength sensitive opsin. It tends to be in the blue. For the red opsin, LWS, these pigments or these photoreceptors match pretty much exactly what you expect. In fact, with LWS, a long wavelength sensitive opsin, based off of 5 amino acid sites that are known to affect spectral tuning, you can predict almost exactly what the spectral tuning should be. And in fact, the alligator and the crocodile are identical to, so I predicted 535, or almost identical to 545 versus 546, what you actually see measured in these taxa. So those identities have been assigned. This one's a little bit more complicated, so you see 566 right here in this double cone member. The only pigment that would make sense for that is LWS, which, like I said, has already been accounted for here and is predicted to be 535. What can happen, though, is sometimes you can get a mix of the vitamin A molecule that's present, and so you can get a mix of vitamin A1 and A2. So perhaps that's happening right here. There's no other pigment that really matches that profile, though, so that's a further question for another day. But what we have left over are these accessory members of these double cones in the alligator and caiman, and then a single cone class that are not accounted for based off the visual pigments that are present in the genomes of these crocodilians. And what I propose is that these are actually our rod pigments expressed in cones. And the basis for this is, one, the only 2 opsins that really come up at 500 to 510 are RH2, which is a gene that I just mentioned was pseudogenized in all 4 crocodilians, and RH1, the rod pigment. Now, beyond that, just in terms of thinking about its spectral identity, is if you compare the spectral sensitivity of the rods to these cones, they're either identical or nearly identical. So a rod here peaks at 501, 503 right here, very close, could be a measurement error. Caiman, 506, 506. Saltwater crocodile, 503, 502, very close. Freshwater crocodile, 510, 510. Okay? So I think this is strong evidence that the rod opsin has been co-opted for cone-specific vision. And as far as I know, this is unprecedented. I've never seen anything in the literature like this. There is an analog in amphibians, though. In amphibians, you actually have a cone opsin expressed in rods. And before that, that was extremely unique. And what this is also suggesting is not only is there a rod pigment potentially being expressed alongside a cone pigment in double cones, which is interesting, but we don't know what double cones are really doing, so it's kind of an open question what that means. But these 2 taxa only have 2 single cones, suggesting dichromatic color vision, so a step below what we have. But potentially by co-opting the rod opsin, what this has done is allowed for regaining of trichromatic color vision. And this is potentially a novel mechanism for regaining trichromacy. So we do actually have other examples of that in vertebrate history. Probably the foremost, and the ones that came to mind when I was doing this at least, are in primates. But they're doing it in a very different way. So in primates, one of the way they've done it is, so in our common ancestor with old world monkeys, they've duplicated one of the genes. And for certain other primates, including some lemurs and New World monkeys, they're heterozygous at the locus at certain spectral tuning sites. And that allows for heterozygotes, at least, to be trichromatic. So this is kind of just interesting to think about the idea of crocodilians potentially regaining trichromatic color vision. Now, I know that's a headache to look at, so I'll go over this quickly. So beyond color vision, not only did I look at the visual opsins, I also looked at other genes associated with light-dependent functions. So here's some examples of some of the genes. I'll go into more detail about them, but I want to point out is just examples of deleterious mutations as well as shared inactivating mutations. So here's that cone opsin I told you that was pseudogenized. Here's the exon, and you notice the crocodilians, it's been totally deleted. There's other examples of frame shifts and premature stop codons beyond that. Here's a gene where you can see in the crocodilians frame shift deletion, frame shift deletion, frame shift insertion, frame shift deletion. Here's some other mutations right here for another gene. So just trust me that those are all light-dependent genes, and now I'll explain to you what they actually do and maybe what's going on for why they might have been lost. So one of them is pinopsin. So pinopsin is expressed in the pineal glands of many vertebrates. So you can see some immunostaining of a pineal gland right here. And so the pineal gland is actually photoreceptive in most vertebrates. And if you actually, it might be difficult to see in the back here, but here's some brains. This little tiny stalk at the top there, that's the pineal gland. Whereas in mammals, it's recessed deep within the brain. And so it's photosensitive, and so there are light pigments in there, including pinopsin. And what's interesting about pinopsin being lost in crocodilians is apparently crocodilians don't have a pineal gland. And I talked about that, I think, at the MVZ, whatever it called, end of the year talk earlier this year. But what this might be, the reason this might have been lost is perhaps when thinking about what the pineal gland is doing is it's actually secreting melatonin and telling the body what time of day it is. So perhaps if you're nocturnally adapted, you lose that function. It's no longer important if you've been dim light adapted for that long. Again, mammals, they actually have a pineal gland, but their pineal gland is recessed deep within the brain. And it's actually lost its photosensitivity, and so similar to crocodilians, they've also lost pinopsin. So this happened in parallel, the two different clades. Another gene that was lost is called Mammal-Like Melanopsin. So melanopsin is found in mammals. It's found in the retina, and what it does, the retina as well as the iris of the eye, and it's important in regulating the pupillary response as well as in trading the circadian rhythm of the brain. And what's interesting about this is this gene is present in mammals, but it's not present in crocodilians. But crocodilians, they have circadian rhythms, they do have a pupillary light response. Well, there's a paralog known as OPN4X, which was discovered relatively recently, and that's still retained in crocodilians. Whereas in mammals, they lost OPN4X, but they retained OPN4M. And you can see they're both expressed in the retina here. This is just some immunostaining. So the question is, why was one lost and not the other in these different lineages? That's an open question. We still don't know a lot about the divergent functions of these. Perhaps they're functionally redundant and it's okay to lose one but not the other. They do have very similar expression profiles. But nonetheless, that's kind of an interesting little divergence there. At least analogously, the same thing happened in mammals and crocodilians. Another one is called neuropsin-like 2. This gene is also lost in mammals. It's completely deleted from crocodilians, confirmed by syntonate. And what's interesting about this is it's an opsin, it's a light-sensitive pigment. It is expressed in the retina and a little bit of the brain as well, but very low levels compared to the adrenal glands. And again, we don't know what this is really doing very much, but the fact that it's expressed in the adrenal glands, you can see here some immunostaining at such high levels, I think it suggested that perhaps since the adrenal glands are involved in sex hormone production, perhaps the adrenals are directly light sensitive. And so for species that have seasonal changes in their reproductive strategies, then perhaps this could be what's going on. But again, the major point here is lost in mammals, lost in crocs. And two more genes that were lost in mammals and lost in crocs are EEVS-like and MTOX. We don't even have official names for these yet. But these were just discovered last year, and what it's been determined is that they function in a catalytic pathway to produce a compound called gadusol. So you can see here how they're kind of contributing to this reaction. What gadusol does, so gadusol is actually quite ubiquitous. You'll see them in bacteria, algae, plants, etc. We didn't know vertebrates had them until very recently. And what it does is it's a UV protective compound. It's a sunscreen. Okay? It was discovered in zebrafish, and it's been shown. So here in yeast, when you irradiate those that don't have gadusol, they have different. They essentially lose. I forgot what this reporter is right here, but they essentially not having gadusol protects these cells from UV radiation. That's the main point. And you can imagine if you're dim light adapted, you're spending all your time in the dark. What's the point of having UV protection mechanisms? So perhaps that's what's going on here. And so I was interested in not just thinking about the fact that these were lost. I also want to think about when these might have been lost. So I used gene inactivation estimates that are essentially going off the assumption that as a gene loses its function, the selection pressure on that gene changes. So we would imagine that across a phylogeny, on average, purifying selection would dominate the selection regime of a particular gene. But as soon as loss of function mutations are introduced, or perhaps just selection becomes relaxed, the gene is no longer important for the fitness of the organism, then that selection regime changes along that branch. So it's essentially taking advantage of that assumption using the NDS ratio analyses. But I didn't just want to map this onto a tree and say, looky, this is when it might have happened. I wanted to think about, are there any correlates in the fossil record that might be indicative of nocturnality or some kind of dim light adaptation on the crocodilian branch? And the one feature that came to mind was the presence or absence of sclerotic rings. So like I told you guys, sclerotic rings are involved in focusing of the eye. And it's not so important in bright light conditions, it is very important in bright light conditions. It's not so important in dim light conditions to actually have that accommodative mechanism. And so what you notice is when you look in the fossil record on some stem crocodilians, such as these guys right here, they do have sclerotic rings. Whereas at least by this guy, Goniophilis, which is one of the earliest, or at least its relatives, not just him, there are no sclerotic rings present in these taxa. And so if you kind of look on this phylogeny here we have crocodilians, here's birds, birds have sclerotic rings, and you could see these fossil taxa right here had sclerotic rings, which are indicated right there, whereas Goniophilidae, they did not. And so we think at least by the early Jurassic, when this lineage appears, that perhaps by then nocturnality or some kind of dim light adaptation was dominating the evolution of these taxa. When you map the actual inactivation of these genes, they all post-date that point. Okay? It seems suggestive of that. I do want to mention that sclerotic rings don't preserve very easily in the fossil record, and so there could be some bias in that, of course. But nonetheless, it is interesting to at least notice this association for now. And one thing to notice I found kind of cool was these two genes, EEVS-like and MTOX, the gadusol enzymes, the estimates for their knockout were almost exactly the same. And these are part of that same gadusol pathway, so it would suggest if you lose one, you lose the other, and that's at least corroborating that idea right there. And so what I want to take away from this is, just like mammals we think went through this nocturnal bottleneck, where they lost a lot of features, this isn't even all of them, but associated with anatomy, and anatomical features associated with bright light vision, as well as various genes associated with light dependency. You see a very similar thing happening in parallel crocodilians, suggesting that perhaps this hypothesis based off of ocular anatomy does have a lot of weight to it when you have the genome telling basically the same story. Now, one thing I want to mention at the end real quick is the loss of two other genes right here. I'm not going to show you in detail, but just look at these and just accept that they're kind of messed up, okay? Lots of frameshift mutations. These genes I didn't find at first, and I had to really, really dig into them to actually find them. But not only are they inactivated in crocodilians, these are opsins that were inactivated not only in crocodilians, I found inactivated mutations in turtles, and I also found them in birds. And there's currently no idea that currently is being held that turtles or birds are ancestrally nocturnal. In fact, birds are overwhelmingly diurnal, and we think that's a very important part of their ecology. So what's going on here? Well, thinking about the function of these genes probably gives some insight as to what's going on. So OPNPT is called Parietopsin, and it's so named because it's found in the parietal eye of lizards. And so the parietal eye is the so-called third eye at the top of the head of squamates. I'll show you guys pictures in a second. But for now, you can see this is Podarcus, and it is immunostained here in these outer segments of the parietal eye. And then also in the iguana, Parietopsin is found in the parietal eye as well. The other gene is called Parapanopsin, and the Parapanopsin was discovered in the parapineal organ of catfish. And for those not aware, which I assume is almost everyone, the parapineal organ is an organ found in some fishes that is para alongside the pineal organ. And they seem to be functioning side by side and doing something with the melatonin response. And what's interesting is Parapanopsin is not only found in there, but it's found in the parietal eye of iguanas, which is interesting because the parapineal organ is thought to be homologous with the parietal eye. Okay? So, again, thinking about the parietal eye, so you can see some examples here, here, The tuatara is famously known for having its third eye. It might be really difficult to see, but maybe you can see this one right here. And it's a photosensitive organ, which is very similar to the eye in morphology, just at a much more degenerate level. So you can see here it has sort of a cornea, sort of a lens. It has a humor right here. It has a retina-like structure. It even has a nerve coming off of it. Okay? And it's thought to be involved in probably doing something to do with thermal behavioral regulation for temperature. But based off of its distribution across extant taxa, so here I just have these skull diagrams, and here you see a hole right there at the head representing the parietal eye. It's only found in lepidosaurs. It's only found in tuataras and some squamates. Crocs don't have them, birds don't have them, turtles don't have them, mammals don't have them. But what's interesting is if you look into the fossil record of these various taxa, the parietal eye leaves the signature of a parietal foramen, so a little hole in the top of the skull, usually found in the parietal, sometimes up to the frontal. And you could see it here in some very early tetrapods, very hard to see there, but you can probably see it in this guy, Campostega. In some stem Archosaurians, there was a parietal eye. In the taxa that we think might have been sister to turtles, you could see evidence of a parietal eye. And in stem synapsids, or in synapsids, stem mammals, you could find a parietal eye. And so we see in parallel, there was this loss of the parietal eye across all these different lineages. And it appears that this happened to co-occur with the loss of these two opsins. So mammals don't have these two genes anymore. Again, at least squamates do. No one's looked at a tuatara yet. But turtles don't, these guys don't, and they all have inactivated mutations on what's left over. The only thing I can't say about this is they were so fragmentary that I found shared inactivating mutations in crocs, I found shared inactivating mutations in birds, I found shared inactivating mutations in turtles. But there wasn't much overlap between all the different taxa. And so they might have been lost in the common ancestor of all of these, or the common ancestor of archosaurs and turtles independently. But the point is, it's difficult to say, and who knows, we might not be able to figure out based off the available information. But nonetheless, I think this is interesting because it's a genomic remnant of a sensory organ that used to be present in all these different taxa. So in conclusion, turtle color vision genes, there's very limited evidence of adaptation at stem lineage. Again, perhaps because all he did was switch that chromophore. That's what I'm thinking at least happened. The descendant lineages do show evidence of adaptation. What those specific adaptations are is difficult to say for now, but at least it's pointing in that direction. Crocodilia, there was massive amounts of gene loss associated with light-dependent genes, and this corresponded with these reductions in ocular anatomy associated with bright light vision. Again, lending support to this nocturnal bottleneck hypothesis. And we think the ancestral crocodilian probably reduces color vision, but then maybe some crocodiles regained a channel, and doing it through perhaps a novel mechanism. And again, the parietal eye loss likely co-occurred with the loss of these two opsins as well. So with that, I just want to acknowledge the following, and thank you so much for your attention. Dr. Muller.
So I'm thinking about the idea that one of the opsins in crocodiles was co-opted from frogs, and wondering if it's moved to like a new protein-protein interaction environment, if there's been positive selection or rapid evolution of that opsin.
Yeah.
Or if you see any evidence of the protein evolving really quickly. There might be another line of evidence that it's being co-opted.
Right, good question. I did not think about to do that, but what I could do is not only check the evolution of the opsin, perhaps also the phototransduction elements to see if something's going on there. That's a good point. And what's interesting about that is, well, it's tricky to say if because you don't see the same expression patterns in the alligatorids, so the caiman and the alligator, we don't know what gharials expression patterns are like. But the crocs, they seem to co-opt it for a single cone, whereas the alligatorids are expressing for that accessory double cone. So perhaps different things are going on, maybe in parallel, or maybe it was ancestral. But the point is, it would be interesting to check both the ancestral lineage and descendant lineages to see if something interesting is going on. It's a good point. Yeah.
There is one turtle that has a parietal eye. I wondered if, or a parietal foramen. I wondered if you would expect them to reacquire any of their opsins.
It's interesting that you know about that. Yeah, so these are gopher tortoises, right?
No, radiata. Well, gopher tortoises too.
Gopher tortoises too. Okay. So yeah. So what's interesting is this was work done by him actually. It's surprising that he knows that. What's interesting is from what I remember looking at what I've seen is the parietal foramen is not like a typical one. It's almost like the fusion of the structures is not happening the same way. So it's not this nice clean circle, which is interesting. Like so, it's like okay, what are they doing there? Another thing actually is leatherback sea turtles. They have, I don't think there's anything going on with the skull, but they do have this lightening of the skull where there's a reduction in the pigmentation, which is also what you see with parietal eyes. There's a reduction in pigmentation. And the thought is that that is performing the same general function. And so in terms of these opsins that are found in the parietal eyes, at least from the few taxa we know, those are so trashed, I can't imagine that would possibly happen. That would be insane to re-evolve that. But still the pineal is photosensitive, and turtles still have pinopsin, as well as perhaps other opsins might be expressed in there. And so that might not, what they might be doing is instead of relying on a parietal eye, they're just having that pineal somehow becoming more photosensitive. Now why those taxa though, that keeps me up at night. I don't know. I've thought about this a lot. It's weird because it's not like it's just tortoise. It's just a few little tortoises and those guys and
the exceptions drive you crazy.
Yeah, and then leatherback sea turtle, not other sea turtles, just leatherback sea turtles. So, I don't know, but it's definitely a possibility.
Chris, you mentioned that there's an opsin expressed in the adrenals.
Yeah.
How much light could possibly be getting into the adrenal gland?
Yeah, good question. So I mean, so the thing about that is, and when we talked about this, I think once in lab meeting, I remember when I first was thinking about the idea that pineals are light sensitive. Brains are also light sensitive, so there are opsins expressed in the brains. Spinal cords are also light sensitive. This is all the stuff we're just starting to really realize. And it's really weird to think about because, again, how much light is actually getting in there? And I can't really comment on adrenal specifically, but the point is we do see this in different body parts we would never expect, first of all. And second of all, we do know there are some modifications to the anatomy that allow for this, at least in some instances. So again, there's no parietal eye in birds. And yet, through feathers, through the skull, through the skin, through other tissues, light is actually getting down there. And that's partially because they have a thinning of the skull. I have no idea if anything's going on with the feathers. I don't think so. I've never seen anything to suggest that. But again, how much light is getting there is an interesting question. But apparently, to some extent, it is.
Has anybody measured that on specimens?
Not that I know.
Could
The only
conceivably
only
do.
thing I've seen is someone has looked at, I forgot which system this is and everything, but someone has actually measured through the skull specifically and see what wavelengths actually reach down into the brain. And they found that the wavelengths that do reach there, sure enough, the opsins present there match that spectral sensitivity almost exactly. So it'd be interesting, obviously, for people to look at this in more detail and think about what's actually going on here. So, yes, Marvel.
Well, actually, there is an old literature that is suggestive of exactly this.
Okay.
And that has to do with the fact that adrenals are in part composed of chromaffin tissue. Chromaffin tissue is dispersed in vertebrates other than mammals and has been thought to be light sensitive. Furthermore, some of the pigment cells in skins have also been thought to be light sensitive. These are all neural crest derivatives, which goes along with your brain, spinal cord, and so on.
That's interesting. Oh, cool.
Yeah.
I like
So
that.
the question is whether or not they're actually still active in adults or if it's just embryonic.
And they're thought to be active in adults.
Okay.
Ancient, yeah, in the 40s and 50s they're
Wow.
I didn't read it when it came out.
1840s to 1850s.
So what wavelengths are getting to these internal sensors? Or?
It's basically cutting off from the extremes and centering around 500s to high 400s, I think, as a rule. There is, and it depends on the organs, but that's the tendency. Exception meaning, I will say, in the parietal eye, there is, is it parapanopsin is actually ultraviolet sensitive. But again, that makes sense because, I mean, you actually have a structure that's allowing for entrance of these wavelengths. Yeah, this is pretty wide open at the moment. We're just starting to really get a handle on how many things are expressing opsins, and pretty much every opsin I just told you guys about, you go 10 years ago, we didn't know they existed. So, there's a lot going on with that.
Well, help me understand this. These are maybe sort of anatomical switches that we aren't aware of, but the body is, I mean,
That's part of
what's the
old
theory
suggestion.
or hypothesis behind these?
There's been no follow-up. This is getting much closer. Yeah, it's all out there.
Okay, well, thank you very much, Chris.